Welcome everyone to the Precision Medicine seminars. So it's my pleasure to introduce Dr. David Knowles today. He's a computational geneticist and a core faculty member at the New York Genome Center. He joined Columbia New York Genome Center in 2019, I believe. And he has a PhD in machine learning from the Cambridge University. He did his postdoc in computer science and then in genetics at Stanford. And here at Columbia, he holds the title of Associate Professor in Computer Science. He's also an affiliate member of the Data Science Institute and interdisciplinary appointee in the Department of Systems Biology. And his research is really focused on applying machine learning-based approaches to the interpretation of non-coding variants and their effects on the transcriptome. So pleasure to have you here.
Thank you for the introduction. Some very interesting background noises. Yeah, so thank you for the invitation. I'm always sad not to make it up here for these talk series. I'm usually at the New York Genome Center, and it's a surprisingly long way uptown to get here. So I always used to be jealous of people having like pretty pictures and feeling like I didn't have a very good way of having pretty pictures in my slide decks. But now we have generative AI, so I get to like illustrate my slides a little bit more. My favorite caveat about this is you'll notice the prompt I had to use for this cute robot analyzing your DNA was a good robot studying DNA to cure human disease. If you don't put the good in, you get something that looks like Terminator and looks very scary. So you have to, yeah, be careful with your prompting. So I'm going to talk about particularly non-coding variant interpretation using machine learning. Everything I'm going to talk about is going to be in the context of Alzheimer's disease, and this is purely driven, really, by the fact that it was one of the first grants I got was to use machine learning models to try and interpret variants in AD. And Alzheimer's disease is, for the most part, a relatively typical complex disease. It's pretty polygenic, it has modest heritability. I like this plot from this 2021 paper where they plot the x-axis here is time, and you can see these different genomic loci being discovered that are associated with Alzheimer's. Just to give you kind of a bit more of an understanding of the plot, the size of the circles here correspond to the odds ratio. So really what basically the effect size. So bigger circle means larger effect size. The y-axis is the minor allele frequency, as with most complex traits. The things that have higher minor allele frequency tend to have smaller effect sizes, and that's because of natural selection. There is the one slightly weird thing about Alzheimer's disease is the existence of the APOE alleles, where you have APOE4, which is this really high minor allele frequency variant that has a really huge effect size. And it's not actually that weird when you think about it more, because Alzheimer's disease, this is all late onset Alzheimer's disease. So this is a disease that's tending to occur, let's say age 65 is a common threshold people take for defining late onset Alzheimer's disease. So it's not having an effect on fecundity. And obviously natural selection, unfortunately for us, doesn't care about variants that increase our risk of diseases that don't reduce our fecundity. So, you might argue that's also why we don't have such brilliant defenses against cancer, because most cancers are happening later in life. The other nice thing about this plot is you'll notice the little boxes around some of the genes, the gene names. And the little box is meant to mean that it is reasonably well understood what the causal gene is in that locus. If there's no box that is saying the name that's given here is just the gene that's nearest to the most significant SNP, that might be the true causal gene in that locus. It might not. And that's one of the things that we're sort of hoping to help figure out. So the reason we got some funding to look at Alzheimer's disease was that the NIH has been funding an enormous amount of whole genome sequencing in Alzheimer's. And so this is under this umbrella of the Alzheimer's disease sequencing project. And this is a timeline that is pretty out of date at this point because there was a pandemic, which you guys may remember, that set things back quite a bit. So the data I'm going to be talking about having used is this R4 release, which has about 36,000 whole genomes in it. We actually just got, like a month ago, this R5 release, which was meant to have 78,000 individuals. It ended up with something more like 56,000, but still like a nice sample size bump. And as you are all aware, we are just at the beginning of 2025. So the pandemic knocked them back about a year and a half and something like 20,000 individuals. They've also been doing, the consortium's been making a heroic effort to do phenotype harmonization. So these big sequencing projects, you can't get 50,000 people from one disease cohort. So to get these big sample sizes, you have to go to different hospital networks across the country, and they're starting even to bring in samples from other countries, even and other continents. And then there's this challenge of, okay, maybe you have case control status. Even that might not mean the same things. We found out recently some of the cohorts in here, when they say it's a case, they really mean it's a dementia case, like broadly defined, rather than Alzheimer's specifically. But there's also all this other phenotype data that in principle you'd like to look at. So for Alzheimer's disease, there are various cognitive phenotypes that you'd like to look at. There's imaging phenotypes, but they always collected a little bit differently across different studies, especially if you're talking about different countries. And so trying to harmonize all that, it's a big challenge, which I'm very happy that we are not the ones doing, but we're hoping to use that data kind of as it becomes available. The ADSP is extremely proud that they finally are at a point where the European ancestry individuals are a minority. There's been a big effort to try and get more ancestrally diverse individuals included in here. And that's gone pretty well for African American, Hispanic. The AMR here means Hispanic, and SAS is South Asian, so that's primarily Indian ancestry individuals. You'll notice EAS is there at 0%. I think it's actually like 0 point something, but there is a big push, and this is for the R4 release. For the R5 release, they are getting a pretty good representation now of East. So the EA is East Asian ancestry. So the work we've been doing in this context is primarily trying to use these deep learning models of gene regulation in Alzheimer's disease relevant cell types to try and interpret the variants coming out of this whole genome sequencing data. So this is a collaboration with Tawfiq Raj at Mount Sinai, and I'm just listing some of the folks involved in doing the actual work. So the big picture idea here is you train these deep learning models to predict different molecular phenotypes from DNA sequence. And so that's going to include both transcription regulation models, so things like DeepC and Informer is one I'll talk about the most, as well as we have been messing around with our own in-house models. So on the transcriptional side, you'd be trying to predict things like chromatin accessibility, transcription factor binding, and so on. And then we're also very interested in post-transcriptional regulation. So this would be things like RNA splicing as one of the molecular phenotypes we've thought about the most. And again, we're going to train those deep learning models using data both from Tawfiq's lab, who generate a lot of data, especially in microglia, which are known to be one of the most relevant cell types for Alzheimer's, and then also publicly available data, multi-omics data for other central nervous system cell types. So your oligodendrocytes, astrocytes, neurons, and so on. So once you have one of these deep learning models that predicts a molecular phenotype of interest from DNA sequence, already for these post-transcriptional models, it would be from the RNA sequence, you can do this thing called in silico mutagenesis. So the idea here is you give the model the reference sequence, and then you get a prediction, and you give it the alternate sequence, and you get a potentially slightly different prediction once you put that variant in. And you can just ask what's the difference between those two predictions. And if there's a bigger difference, that implies that the variant has potentially some functional effect according to your model. The really nice thing about this is the model was trained to generalize across the genome. So I trained a model, let's say Informer, or I didn't train, Dave Kelly trained this model to generalize across the genome, across different genomic regions. And from that, I am then able to interpret basically any genetic variant that you give me. So I don't have to have seen this genetic variant before. And that's kind of the big selling point of these methods is I can interpret any variant. It could be super rare. It could even be a de novo mutation that no one's ever had before. So we call those resulting variant effect predictions delta scores. And we're using them in various statistical genetics analyses. For example, trying to do functional find mapping. So this is in a genome-wide association study, can you figure out what are the actual causal variants? As those of you who do anything with GWAS will know, GWAS gives you association. It doesn't tell you anything about which are actually the causal variants. And so find mapping tries to do that, and you can try and incorporate this sort of information about likely functional effects of variants to try to do that better. Partly to test the fine mapping and partly because it's potentially of interest in its own right, we've been trying to see if getting the causal variants can help give you a more portable polygenic risk score. So polygenic risk scores are basically saying, can I predict, in this case, Alzheimer's disease risk from your genotype data? And it's been observed that if you try and do that using European GWAS data and then apply that to, say, individuals of African ancestry, you get horrible predictions, like basically better than random. And so this is potentially one way of improving that, and I'll talk about why that potentially should help when I get to that section. And then we're also using these variant effect predictions to try to do better non-coding rare variant association studies. So the idea here is that we would like to use rare variant burden to prioritize additional genes that seem to be involved in AD. But from whole genome sequencing, you get a lot of variants, especially if you're considering non-coding variants as well. So it's sort of the needle in the haystack problem. You expect that most variants aren't doing anything, and you'd hope that these deep learning models can help tell you which variants are actually likely to be having some functional effect, and you can upweight those. So yeah, the Informer model, which was published, well, I guess that counts as 4 years ago now. It's a collaboration between Dave Kelly's group at Calico and folks at Google DeepMind. And it was the first time that people had really applied a sort of transformer architecture in the context of this setting of trying to predict molecular phenotypes from DNA sequence. They didn't use a pure transformer architecture, which I think makes a lot of sense. They use these convolutional layers, which have been kind of the bread and butter of these sorts of models up until that point. The convolutional model you can think of as scanning something like a precision weight matrix. So basically a thing that looks for where might there be transcription factor binding motifs along the sequence. And then the transformer components, which are, the basis of all of these generative AI models that there's so much excitement around, your chat GPTs and so on. They can do very complex reasoning about the long-range interaction between these different binding signatures. They train this on a ton of data from both human and mouse. So you can see over 5,000 different chromatin marks. This is everything you could possibly get your hands on: chromatin accessibility, transcription factor binding, all those sorts of things. A lot of this coming from these big consortium efforts like Epigenetics Roadmap and ENCODE that have spent, basically the 2 decades since the human genome was kind of drafted, at least, generating all this annotation data. So the Informer model you can use to get variant effect predictions. One of the caveats is there are lots of cell types of interest that are not well represented, even in those 5,000 tracks. And so one of the things we've been messing around with and found, at least to my mind, surprisingly challenging, is saying, could we take the existing Informer model and fine tune on cell types of interest? So for example, I mentioned microglia as being very important in known to be very important in Alzheimer's disease. So we have multi-omics data from microglia. This turns out to be challenging. I think the setups we have for trying to fine tune, so trying to adapt these existing models to new data, are not so well figured out in this setting compared to in natural language processing or in computer vision settings. And also, I was talking to some of the trainees earlier about this. There's potentially some data quality issues as well. So microglia are very fragile cells. They are typically studied by try to sort them from postmortem brains, and they don't like that process. And the data quality you get is lower than what you'd get from cell line data. So there's this trade-off of, okay, you've got the right cell type, but the data quality is maybe not as good as it was for related cell types. So another approach that we've been taking that for now has proved more profitable has been to say we can use the deep learning model to get predictions of which variants would be disrupting transcription factor binding. And then we can overlap that with multi-omics data from the different CNS cell types that lets us define CREs, that's cis regulatory elements, i.e. promoters and enhancers. And this is all work done by my staff scientist, Chirag. So the idea here we borrowed from Bryce's paper. He was a postdoc. We overlapped at Stanford, and then he was a postdoc with August Bryce. And this is his postdoc paper where they propose this idea. So they're basically saying you can use the deep learning-based predictions of transcription factor disruption, overlap that with enhancers and promoters from your cell type of interest, and use those as annotations for downstream tasks. So this is showing you what you get if you do that, and then you relate that to those variant annotations to Alzheimer's disease GWAS. This is using an approach called stratified LD score regression. So the three different rows here correspond to different Alzheimer's disease GWAS. I'm not going to go into the details of these, but the Alzheimer's disease GWAS are done in slightly different ways. One big difference, for example, being whether you use disease by proxy cases. So if you look at UK Biobank, for example, you won't have many individuals who have Alzheimer's disease because it's not an ascertained population and the typical age is sort of middle-aged. But you can ask if people's parents had Alzheimer's disease, and people are obviously related to their parents. So you can use that indirect information to get some more signal for your GWAS. It's a bit controversial how well that actually works. So that's the different rows, the different columns here. So the first column is showing you the proportion of SNPs that are in the cis regulatory elements for each of the cell types, which are the different colors, and are also predicted to disrupt transcription factor binding according to the informer model. You can see, interestingly, actually a bunch more in neurons. I don't know if that's a real biological difference. I think it potentially could be. There's actually a big diversity of neuronal subtypes that are represented in that, broad cell type definition of neurons. So it could potentially be a real thing. It could also be something to do with the data. And then the interesting columns are the middle and the right. So the middle is showing you the proportion of heritability. So how much of the GWAS signal is captured if I just consider SNPs in that annotation. And so you'll see that the number is higher always for microglia in red. And if you look at Kunkel, for example, at the bottom, which is maybe the cleanest GWAS, it doesn't use that disease by proxy approach that I was telling you about. You can see, okay, there's a very wide confidence interval, but the middle of that confidence interval is a bit over 50%. And so what's that? Let's say that's 58%. So that would be saying 58% of the signal in the GWAS is captured if I just consider variants that are in cis-regulatory elements of microglia and are predicted to disrupt transcription factor binding. So that's, I think, a pretty remarkable amount, right? Like there's lots of other things going on. There's other cell types, and maybe we don't have a perfect definition of cis-regulatory elements, but you are seeing that, like, even given those caveats, a lot of the signal of the GWAS is getting captured if I just look at microglial regulatory elements. And then the rightmost column is showing enrichment of heritability. So this is saying one way of interpreting that the numbers on the x-axis there is how many times more heritability is captured by a variant in that annotation compared to like a control variant that's not in that annotation. So a number that, so say for the microglia here, you're getting a number around 12. That is saying the typical SNP, which is in a microglial cis-regulatory element, and we predict disrupt transcription factor binding, carries 12 times more signal than a variant which is not in that annotation. So very strong enrichment in microglia, and this is consistent with previous studies. You don't just need to limit to those annotations. You can look, you can do this sort of LD score regression-based analysis for a much wider range of these functional annotations, and that's what I'm showing here. So the x-axis is again this heritability enrichment score. And the different categories here, the different colors. So the blue here corresponds to variant effect predictions directly from an informer. And you can see actually the strongest enrichments you get are for monocytes, for DNAs, which is a way of measuring chromatin accessibility, and for H3K27 acetylation, which is a measure of regulatory element activity. This ZS3, this is the annotations that Chirag makes. He uses different Z-score cutoff. So the 3 is the most stringent cutoff for the variants having the biggest effect on those chromatin marks. Monocytes make sense, it turns out, because monocytes and microglia are very closely related cell types. So monocytes are the innate immune myeloid cells that are circulating in your blood, whereas microglia are the equivalent population that infiltrated into your central nervous system at some point during development. And so monocytes sort of acts as this proxy cell type that, for one thing, you can get better quality data on, and for another thing, was actually in the training data set that Informa was trained on. And so it's very valuable to us here. You know, in an ideal world, we would have figured out how to train an equivalent model to Informa or fine-tune Informa for microglia, and it would outperform this. So far, we've not managed to do that. Still trying. And you can see the next feature that comes up are these microglia. This EP is enhancer and promoters intersected with attack-seek, it's the A in yellow. And then this next in green, this is the sort of feature I was telling you about here where you're overlapping things. So all of these features contribute. You can see all of the top 5 features that annotations that come up as the most associated are all to do with microglial regulation, which makes a lot of sense. And then you start to see the new N is neurons. You start to see some neuronal signal a little further down as well. So this is interesting from the sort of, it makes sense from the known biology of AD. But we wanted to ask if you can use that information about the types of annotations that the variants that are the most strongly associated are in to do a better job of this functional fine mapping task I was telling you about. We are trying to figure out what are the causal variants. And so there's this nice approach called polyfun, which combines stratified LD score regression, which is the results I've been showing you, with a fine mapping approach called SUSY from Matthew Stevens' lab. So SUSY, it's a really elegant Bayesian model fit using variational Bayesian inference to try to do this fine mapping task of figuring out what are the causal variants. One of the nice things about it is it lets you find multiple causal variants, independent causal variants in a locus, and it's computationally efficient. So I'm showing one example of applying functional fine mapping to this quite well studied PyCharm locus. So this is what's called a locus zoom plot. So here you've got the y-axis here is the minus log 10 p-value from the GWAS. And the x-axis is showing this genomic locus where there's this PICAM gene and this EED gene. And you can see there's these SNPs that have the strongest association here, but there's also a lot of SNPs in strong LD, that's linkage disequilibrium, so highly genetic variants whose genotypes are highly correlated with these SNPs. So it's one of these regions that's really tricky to figure out like what's the likely causal variant. And I'm showing you a zoom in here. You can see these two SNPs are in almost perfect LD, so perfectly correlated, and have, as a result, basically exactly the same p-value from the GWAS. And if you just do fine mapping without using any functional annotations, any of these variant effect predictions, then you can see the SUSY result here. There is a tiny bit of difference in reality between these p-values, and the LD structure means that SUSY puts a slightly more weight on this first SNP as being causal. But once you include the functional annotations, then you get much more confidence that this first SNP really is the causal variant, so that the posterior probability of this being causal goes up to 0.9. The signal that the model is using for that is that that first variant is in a microglial attack peak, so in a chromatin accessible region, and is predicted to disrupt transcription factor binding of PU1, which is one of the major transcription factors that defines the myeloid lineage. So this all sort of makes sense. I'm not showing it here, but you can also overlap this with EQTL data. And it looks like it's actually the PICAM gene, not the EED gene, which is the likely causal gene in this locus. So we've done this functional fine mapping genome wide. And once you've done that, you can take the likely causal variants and see if you can do get better polygenic risk scores. So the motivation for this is, as I said, when you take a polygenic risk score, so attempting to predict case control status from genotype, if effectively your training data, your GWAS is European, then it has been repeatedly shown, Alicia Martin was one of the people to really try this point at home, that when you take that predictive model and apply it in a different ancestry group, you tend to get horrible performance. And there are likely many reasons for this: environmental, socioeconomic, and so on. One of the likely genetic reasons is we know that linkage disequilibrium structure changes. So the correlation between SNPs is different across different ancestries. So if you think of African ancestry individuals, for example, we know that haplotype blocks are shorter in African ancestry individuals. And that's partly because of this idea that everyone who is not African went through a bottleneck during the Out of Africa events. And as a result, we have these longer haplotype blocks. So the reason that's relevant is if I'm trying to make a prediction in Europeans, if I don't get the causal variant when I'm making my prediction, if I just get a variant that is tagging the causal variant, that's fine, because the correlation structure in my test set of European individuals is going to be the same as it was in my GWAS. And so that tagging variant is still going to be predictive. But now if I go to African ancestry individuals, that tagging variant might not be correlated with the causal variant anymore. So when I use that to make a prediction, it's not useful. And so the argument is that if I can get the causal variants, my PRS should port better to different ancestries. And we do see this, but it is a modest effect. And I think our general experience with PRS is it remains like a tricky area. But you can see here the x-axis is the area under the ROC curve, so a measure of predictive accuracy. In Europeans, unsurprisingly, you do by far the best. And even in Europeans, you do see some gain. So SUSY here is doing fine mapping without any annotations. And then if we use baseline annotations, so this is a set of sort of generic annotations that LD score regression provides you with, things that include stuff like conservation and some generic chromatin accessibility averaged across many cell types, that sort of thing. Then we do a little better we do a little bit still if we include our multi-omics data across different cell types that are AD relevant. And we do a tiny bit better, maybe again, if we include the deep learning annotations. If you go to African or Hispanic individuals, then the performance overall drops a lot, but you still see this trend that you do better when you incorporate the functional annotations. It looks stronger in the Hispanic ancestry individuals. I don't honestly know if this is real. The sample size for the Hispanic population is smaller than for the African population. So that was all thinking about transcriptional regulation. So transcription factor binding, chromatin accessibility, etc. We have a long-running interest in RNA splicing variation. And so we wanted to think about whether there might be splice disrupting variants that were contributing to AD. And we're taking a similar approach here, but adapting a model called Pangolin, which was published in 2022 by my friend Yang Li in Chicago. So they took the Splice AI model that Yang and I were both involved in co-developing when we were back at Stanford with Illumina. And Splice AI is not trained on cell type specific information at all. It's just trained on gen code to predict splice sites. So what Yang's group did with Pangolin was to train on using RNA sequencing data from different species and different tissues. And the basic architecture stays the same as Splice AI. It's a big convolutional neural network. And it's going to try and predict for every nucleotide in a genomic region, will this be a splice site or not in a given tissue? So what Tatsu, who's a joint postdoc between my group and Tawfiq Raj's group, did was to take Pangolin and then fine tune it for microglia. And this he did get to work pretty nicely. So you can see the accuracy here, so the error under the ROC curve. So blue is making predictions using the original Pangolin model, so not fine-tuned for microglia. Then if we fine-tune with human microglia data, we get a little bump in performance. If we train a model, so the modified outputs here is the Pangolin model just predicts splice site or not. And the modified outputs version here that Tatsu came up with, instead of predicting splice site or not, it predicts 3' splice site, 5' splice site, or neither. So it's like a three-way, which is actually how the original Splice AI worked. And you can see that gives you another bump in performance. And then the best performance you could get was training this model using both human and multiple species data. And I think this is actually an interesting general point for these models, that the sample size when you're training these deep learning models is really the number of different genomic loci you get to see, not the number of individuals that you have. And so having multiple species data is actually really valuable for training these models. And I think this is still pretty unappreciated by funding bodies, that for this sort of modeling, having data from many species is very valuable. Certainly when we've talked to NIH about this, I feel like they don't understand this aspect. They're always asking us like, okay, do you need more samples in terms of more individuals? And for this aspect of it, that doesn't really help. So then you can play a similar game to the in silico mutagenesis we were doing for Informer by asking whether a specific variant will change the likelihood of positions being a splice site or not. You can also combine this with predictions for RBP binding. So we're using a model called SeqWeaver from Olga Transkaya's lab to get predictions of variant effects on RBP binding. And then we're incorporating additional features like the distance from the splice site to train a random forest classifier, which we're calling the splicing modifier score, so SMS, which tries to predict whether a variant will be splice disrupting or not. And we do that. The training data we use for this is fine mapped splicing QTLs. So splicing QTLs are basically looking for associations between genotype and splicing outcomes. You can fine map those to try and get an estimate of which are the causal variants. You can use that as training data for this model. And combining these different features of the fine-tuned pangolin predictions and these outputs from SeqWeaver and these other features like distance from the splice site, you can get reasonably good prediction of which variants will disrupt splicing. Tatsu actually has slightly better performance than this now by adding a couple more features and a bit more like hyperparameter tuning of the random forest model. Um, So I mentioned one of the things we want to do also with these variant effect predictions is to empower rare variant association studies. And this is work that's been led by my PhD student, CS PhD student, Anjali. So she's taking the whole genome sequencing data from ADSP, doing these cell type specific rare variant association tests using defining the set of variants to test using various multi-omics data, and then incorporating these sort of functional annotations I've been telling you about, so deep learning and Bayes and others. So we've been doing some methods development in this space, building off some existing methods that some of you will be very aware of. So there's a whole line of these rare variant association tests, and I think one of the sort of de facto methodologies now is this SCAT-O test, which incorporates both what's called a burden test and a dispersion test. So a burden test sort of making the assumption that every variant you're testing is having the same effect within a gene. The dispersion test allows that there are different variants within a gene can have different effects. SCAT-O, however, does not use variant annotation, so these sort of functional annotations I've been talking about. There have been some more recent methods like FST from Juliana's group and STAR, which do use variant annotations, but they just fit a model for every gene, every set of SNPs separately. So they can't learn genome-wide in the way that LD score regression was for common variants, which annotations actually seem to be important. So we have a model called GRIA. I'll show you the backronym next, that tries to do this. So uses variant annotations and then also learns which ones are important across the whole genome. So we're particularly interested in the non-coding space, although we do also look at coding variants. So we need some way of saying for a given gene, what are the variants that we should consider in our gene set? And so to do this, we're using this ABC model, activity by contact model from Jesse Ingritz's group at Stanford. So they have what's really a relatively simple approach. It uses high-C chromatin looping information combined with chromatin accessibility and histone modification data to try and give you an estimate of what are the enhancers and promoters for a given gene. It is windy out there. And so we use this to link enhancers and therefore variants, including rare variants, to each gene. And then we feed those variants into the various association tests that we're going to do. With the models that use functional annotations, we can also include those after calculating them for all of the variants. So some of the first analysis Anjali did was to look at whether using these functional annotations does seem to increase signal. So this is showing you the difference between using SCAT-O, which doesn't use any functional annotations, and FST, which does. And you can see in general, you do get a boost. You can look at it you can see this just from there's more p-values piling up near zero for FST, which uses annotations versus SCAT. And you can also see it if you just do a scatter plot of the minus log 10 p-values as well. So that sort of motivates using the functional annotations. But as I said, we wanted to try and improve on FST by learning genome-wide which annotations are actually important. So I think this is one of the only two, right? Oh no, there's going to be one more slide that has math on it. Okay, there's going to be 3 slides with math on it. So for those who like seeing the math, enjoy it. For those who don't, I'll do my best. So the basis of GRIA, so genome-wide rare variant enrichment evaluation, it's not too bad. It's better than like if you try and get ChatGPT to do backronyms, it's horrible. So I think relative to that, it's pretty good. So it's a hierarchy of generalized linear models. So each of these generalized linear models is really like a logistic regression for each gene. And it's a hierarchy because what gets shared across the different genes is this vector tau, which basically tries to tell you which annotations, which of these variant annotations are important. So sorry, let me talk this through first. So Y here is going to be case control status. Does this individual eye have Alzheimer's disease or not? And that's conditional on the genotypes of a gene G for that individual, and also some covariates. So these covariates would be things like sex, age, APOE status. We also include genotype PCs in here as well, because we want to control for ancestry. Then there's a regression coefficient alpha for those covariates, then the genotypes, and a regression coefficient beta for all of the SNPs for that gene. So the interesting part of the modeling comes in how we get this beta. So in Gruyere, beta is a deterministic function of these various things. So the J here indexes the specific variant. WG is a gene weight. So this is actually the thing we're going to use for statistical testing, is asking whether WG is non-zero. WJ is a weight for the variant. And this basically weights rare variants to have a higher weight, because we know because of selection, rarer variants are more likely to have larger effect sizes. These Z are annotations. So this is for gene G, variant J, and annotation K. What is the value of that annotation? And the way we have this set up, these are positive and they don't have to be binary. We normally scale so they're between 0 and 1. So this will be, you'll see the sort of things that are included in here, but this can be things like conservation, deep learning-based variant effect predictions, and so on. And then these taus, these are annotation weights. So these weight how important those different annotations are. There's also a tau 0 because you might still have signal even if there's no variant annotation. Any questions on the model? It's tough doing math at quarter to 5. So okay. So this is the set of annotations that Angie puts in the model in the end. I won't go into details of it. She ends up summarizing a kind of much larger set of annotations down to these sort of simplified versions. For example, yeah, please, yeah.
So
No.
From the model you used, what is the known and what's the unknown bit?
Oh yeah, that's a good question. So the known things here would be X and G, so the covariates and the genotypes. The unknowns are alpha and beta. But bear in mind, beta is a deterministic function of these things. So the actual parameters you're fitting are WG, which is effectively gene importance. And the WJ is actually, it's fixed to be this, just copying FST. And then the annotation Z are known. And so then we're tau, the tau K and tau 0 are the other unknowns. And the thing I meant to write on here and I didn't is we fit this using, it's Bayesian-ish, so we fit it using stochastic variational inference in Pyro, which is this probabilistic programming language that builds on top of PyTorch. So you get all the like GPU speed up, which is nice, but you can fit these models in the sort of Bayesian framework and then use that uncertainty estimates to do statistical testing. So the different colors here correspond to the different test sets we do. So for the non-coding variants, we have these different cell types, and then you have different promoter and enhancer regions from ABC defining the gene sets. And then we also have coding here as a separate category, the salmon, I guess. So as you might guess, for coding variants, the most important annotation is this variant predicted to be a loss of function variant. Splicing also comes out, which is interesting, I think. So these variants that we're thinking of as like, oh, this might affect the protein sequence. If it's also predicted to disrupt splicing, and that's based on actually it's on Splice AI, at some point we'll incorporate Tatsu stuff. Then that also has a big effect. And then for the different cell types, you get so these are these delta scores coming from Informer. We took a simple approach here where we just take a min or a max over the delta scores. So we summarize across multiple transcription factors. The other delta scores that come out as informative are these different histone modifications and chromatin accessibility, which we included for monocytes, since we knew microglia were going to be important. So this all looks pretty reasonable. I guess I should say also, Anjali did a lot of work testing the model on simulations and with permutations and that sort of thing, which I'm not going to show. So in terms of results, we see this is sort of the Manhattan plot equivalent for the rare variant association testing. So every point is a gene, the different colors of the different cell types plus coding. The strongest association we see is for TREM2, which is a very well-established AD risk gene. APOE is not tested here because the signal blows out everything else. And then this is showing overlap of GRIA with other methods. And we actually see relatively limited overlap, which I think is interesting. It's a very different approach to other methods because we're really leveraging the functional annotations very, very heavily. And the red here correspond to microglia, which are likely some of the more interesting associations. We were, however, a little bit curious that the overlap with other methods was so limited. And so the next thing we did was to think about whether we could incorporate this dispersion idea that's used in many of these other rare variant association tests. So the GRIA model, the beta, the effect of a variant, is a deterministic function of the annotations. And that's clearly somewhat restrictive. And so what Parmigiano, if you can try and read it off, if it see if it works for you, does is to relax that constraint somewhat. And the way this is done, you can think of as extending that generalized linear model to a generalized linear mixed model. So what that means is the mean of the regression coefficient for each variant is effectively the same as we use in GRIA, but we also now allow there to be variance around that mean. The details of the model construction here are not super important. There's a dispersion parameter rho, which balances how much this mean versus variance component are used. So if rho is 1, you basically recover the GRIA model, whereas if rho is 0, you get a pure dispersion model. And this lambda is a shared estimate of how important each variant is that you'll notice has basically the same functional form, same dependence on the variant effect annotations as GRIA did. We tried having separate predictions of variant importance for the mean and the variance, and that model never fit very well. So the results for Parmigiano are more consistent with other, maybe I'll show this first, are more consistent with other methods. We get similar annotation importance results. And now, actually, interestingly, the strongest associations we get, this REAP3 association, is in microglia. This is a ER stress response gene. So interesting to think about whether that implies there's some stress response component to what's going wrong in microglia during AD progression. So I'll go through this section of stuff quickly since it's 10 minutes to go, maybe just try and give you a sense of this since I want to change gears a little bit. So we've been interested in, the work I've told you about so far is really sort of using the, whatever you'd say, 2015 deep learning excitement. And these generative AI models that are all the rage now, from your ChatGPTs to your Stable Diffusions, are these foundation and large language models. And there are many people trying to apply these models in the context of biology. So as you know, this has worked, these models have worked incredibly well for natural language, right? Like you basically do this fill in the gap task or predict the next token task on internet scale data, and you get this amazing emerging, like almost human-like intelligence. That same idea has been applied in the protein space, most famously by the ESM set of models, and has again been very successful. Just substitute words for amino acids. And ESM fold is one very nice example of this working really well. So AlphaFold needs a multiple sequence alignment. Sometimes you don't have a deep multiple sequence alignment. Maybe you're doing synthetic biology and there is no sequence alignment because you've just made up a new protein. Well, ESM lets you not rely on a multiple sequence alignment and just get an embedding for your protein anyway. And you get very close to the performance, not quite the same performance as you do with AlphaFold. Whereas if you take AlphaFold and don't give it an MSA, the performance really drops off a cliff. In genomics, it's been a much more mixed story trying to apply these things. So we're very interested in this idea of can you apply these modeling ideas to DNA sequence? And there's there's many more papers I'm sure I'm not listing here that have tried to do this. Sometimes they do this at the nucleotide level. Sometimes they look at sixmers, train on multiple genomes. It's also been done for single cell RNA sequencing. It's a bit more of a stretch here. You have to think about representing a single cell as like a sentence that is the top most overexpressed genes in that individual cell. And then the game is, can you like fill in the missing gene out of that list? And again, there's multiple models doing this in slightly different ways. But every time these methods have been independently assessed, so this is a nice paper from Peter Koo, they come up kind of lacking. So this is evaluating these DNA language models and basically showing you get better performance if you just do supervised training, none of this unsupervised or self-supervised pre-training, which is the orange methods here. This is on predicting which variants will be functional in a big MPRA study. This is a nice, very comprehensive paper from folks from Copenhagen showing basically the same story. So maybe to actually show you what's happening here. So this is different tasks. These columns are different tasks. The expert method is basically an existing regular supervised deep learning approach, whereas the pre-trained is trying to use these large language model type approaches. And for the majority of these tasks, you do better with one of these existing supervised methods. Variant effect prediction for saying which variants will be pathogenic, you actually do really well. Nucleotide transformer, this is MS here, stands for multiple species, sorry. So this is one of the biggest models trained on a ton of data, and it does actually do pretty well. So that's sort of motivating. Yeah, that's the conclusion. I will skip this in the name of time, but the story is sort of similar for single cell models. So why do large language models work for text and protein, but not for DNA and single cell data? I think one thing is training data scale. For text, you have the entire internet. For proteins, there are a lot of proteins that we know the sequence for, even if we don't have other labels. Then there's an aspect of information density. So much DNA sequence is not conserved. And one way of looking at that is to look at perplexity. So perplexity is if I'm trying to predict the next token in the sequence, I can ask what if I had k possible tokens that were uniformly equally likely, I can, when I actually make the prediction, what's the entropy of that prediction? Yeah. And this is always hard to explain. Okay. So I'm making a prediction of what the next token is going to be. That distribution has some entropy. And entropy is a really hard number to interpret. However, I can ask, if I had a uniform distribution over k tokens, what is k such that the entropy of that uniform distribution is the same as the entropy of the actual distribution I have? It's hard to think about entropy, but it's easy to think about like I would be uniformly picking between k tokens. Well, perplexity is that k. So that's maybe concretely for English. If I'm talking about words, if I don't have a model, I just know the frequency of words in English, then it turns out the amount of the equivalent entropy distribution is the uniform distribution on 3,700 words. Now, if I have a state-of-the-art model, so I think this is ChatGPT 3.5, state-of-the-art-ish, then I get down to, it's much fewer words that are likely to occur next in the sentence. And on average, there are 10 possible words, is one way of thinking about this. So you can think about the improvement factor here. I went from, I don't get to see, you're just going to say a random word, and I have no idea what that word's going to be. And then if I've seen a bunch of text and I'm saying, what's the next word? the improvement factor, like how much better I would be at predicting the next token is the ratio of these two, so 370. If I do this, think about character level, then that improvement factor is something like 8. Now, if I think about proteins, it turns out that number is quite a bit lower, something like 3. And it's a really small number if you look at the human genome. So the state-of-the-art models for predicting what's the next token, next nucleotide in the human genome, you only get this 1.5 times improvement in versus random. So information density is much lower. Single cell profiling data is noisy. And then I think there's this other aspect that in the protein and text settings, it seems like it's been okay to model those modalities on their own. I think for biological systems, just thinking about the DNA or the RNA or the protein on its own is probably very limiting. Where I'm out of time, so I will just say what we're trying to do is context-dependent DNA language models. So conditioning these models on species, chromatin, and cell type. And I will skip the architecture and just say that when you include this context, you can do quite a bit better. It's still the next thing we want to do is try and use this for the variant effect prediction task. Okay, and I will wrap up there with some concluding thoughts, which is just to say, I think machine learning has this huge potential in variant interpretation. We do see some positive results with that already. There's a lot of challenges as well. And I think one of the ways forward is probably linking across more of these modalities. And thanks to funding and Chirag, Tatsu, Anjali, and Teresa who did the work that I talked about. Okay, thanks. Sorry for being over time. If anyone has whatever that is, 30-second question. Yeah, I hope.
So if these variants in microglia, they are also some of those very common with monocytes. I mean,
yeah, definitely, yeah. Yeah.
You see that some of these risk variants are also associated with other diseases, immune diseases?
Yeah, that's a good question. We haven't done much looking at like cross disease. That would be interesting to look at. I mean, for also maybe potentially for other neurodegenerative diseases. I guess I don't know well enough because the other diseases, the other immune diseases that I see people talking about a lot, you know, your diabetes and these sorts of things are more on the like seem often to be the T cell type, you know, the adaptive immune system, right? So yeah, it'd be interesting to think about whether there are other diseases that are to do with the innate immune system being dysregulated. It's also, I mean, I think it's really interesting in general with Alzheimer's that the understanding of how Alzheimer's progression happens. Microglia are important, but they're important they're not meant to be really involved in disease, the initial disease onset. It's meant to be more of a response thing. Like how are that microglia responding to amyloid beta plaques? How are they responding to tauopathy later on? And so I think that aspect's really interesting, right? You look at the genetics and it really points to microglia. But everything that's at least people say about how disease progression actually goes is, you know, maybe there's some astrocyte stuff going on really early on. And then there's this amyloid beta buildup, and then microglia get involved. So I think that's interesting, right? And a bit confusing of what exactly is going on there. Maybe it's just that like everyone has, you know, a little bit of amyloid beta plaque going on. And it's just if your microglia aren't very good, then you get Alzheimer's disease. It's like everyone has that risk, and you need the microglia there to stop it.
It was this, I remember reading at some point, there was this epidemiological data of people receiving vaccines for chickenpox or something. They were kind of protected for.
Interesting.
I mean,
yeah. Okay.
I don't know if there is any.
Yeah. I don't. Yeah. Okay. Yeah. Yeah. Interesting. Yeah.
For non-coding regions, would you remove coding variants altogether so that it doesn't like override?
Yeah, no, it's a great question. So for this, we do. I mean, the annotations are all like slightly imperfect, but in principle, yes, we are removing that. So it's an interesting question of like what are you trying to show, right? So I guess we're partly trying to show that we can pick up rare non-coding signal, in which case then you can be like, well, if you include the coding signal, you don't know if everything's just coming from coding. But if you just want to have as much power as possible to find associations, you probably want to include both. And actually, I didn't show the results from the splicing variant effect prediction, but you're actually pretty underpowered. It's basically the variants. We think there are some variants that have a strong effect on splicing, but they're pretty rare because they have to be quite close to splice sites to have that effect. And so the issue we're having, if you look at them on their own, there aren't enough per gene to get significance. And so we're trying to look at those combined with, so you're basically asking like, if I did a coding test, coding variant association, rare association test, versus if I do coding plus these predicted splice variants. So then it's sort of like trying to ask is, can you gain a bit of power by adding those predicted splice variants? Because we have this bigger sample size now, this R5 release, so we're sort of hoping that's also obviously a good cheat code is just get more samples. So, good question. Yeah.
Quick question is that Genome now has the non-coding constraint.
Mm.
That you might have, right? Like the 1,000 base pair windows that.
Yeah, right. I don't think we've used that. Yeah, that would be interesting to include. Yeah.
Yeah, I'm just wondering because it might be that some of these highly rare variants can also corroborate like their coding constraint, but I don't know, it can be a noisy estimate because it's a pretty big window for them.
Yeah, but it'd be interesting to see if the model picks up on it. Yeah, that's all. Yeah. But and then also there's the caveat of like, do we expect the Alzheimer's is not under so much constraints, but so like how much it's a good thought. Yeah.
Yeah. So as you mentioned, the substance that the relationships vary by ancestries.
Yeah.
So I'm wondering if the ancestry component of your input data will
Right.
affect the final result, or if they can get the ancestry specific annotation.
Yeah, so I think for better or worse, we are making the assumption that the variant effects will be similar across, but at least the variant to molecular phenotype effects will be very similar across ancestry groups. Whether the molecular phenotype to disease risk connection is the same across ancestries, I think that's more questionable. But the basic, like, if you have this variant and will the functional effect be the same across ancestries? I'm reasonably confident on that because I don't think I don't think gene regulation is going to be different across different ancestries. Variant allele frequencies totally will be different. LD patterns will be different. And then potentially, so it depends what you're conditioning on. So if you're conditioning on this variant existing in both populations, we think most of the time that will be the same. But like the biggest difference you'll see is that the allele frequencies are different, especially for rare stuff, right? Like you're going to have ancestry-specific rare variants. But I think for common variants, it's more the allele frequencies tend to be more similar, I guess. But yes, for now, we're mostly assuming that the variant effects will be the same, which you can argue with. The main issue is we don't have power, right? Like we're already limited on power. So then if we don't make that assumption, I think we're in a pretty tough spot. So I mentioned it briefly, but we're controlling for ancestry primarily using genotype PCs. Yeah.
What do you think will be a good sample size to conduct such?
Yeah, I don't know. I think you want to be in the like tens of thousands per population is my sense. Yeah.
But you don't need other omics as well.
Well, the models can be trained. I mean, the models, you don't need that many individuals. Yeah, right. Yeah.
My understanding is like garbage in goes garbage out.
Yeah, yes, totally. Yeah.
Do QC filtering if like it passes for one omic, but not the other?
Right. No, that's a great question. I mean, we're trying to do this. I mean, I think it was tougher for Informa, right? They're training on 5,000 different tracks. So that's a bit more difficult. For us, we're trying to train on, maybe there's like 20 tracks we want to train on. So it's a bit easier to look at them manually. The bigger thing we're trying to deal with carefully on the garbage in, garbage out issue is these biases that are kind of fundamental to these different modalities. So AttackSeek, for example, Andrew Conjade has done a lot of nice work on this. It has this like enzymatic bias of where it tends to cut. And so he has this approach where you try and train a bias model, which is it's like a simpler deep learning model that tries to predict from sequence the kind of background signal that you're going to get, and then models the observed signal as like that plus the like real biological signal. And yeah, Turing's actually, that's the most recent thing he's been working on, is trying to like improve that methodology. It's tricky because it relates to how you assess the methods. So bias correction like that is an interesting one, because if you just want to say, you always see in these deep learning models, like, oh, we held out these chromosomes and we trained on all the other chromosomes, and then we predicted on like chromosome 20 and 21, let's say. If you don't bias correct, you might actually do better, because you're also predicting that bias, right? So you learn the biases of the assay really well, and you can predict them, and they'll still be there on chromosomes 20 and 21, but that's not the actual biology. So what we are trying to do is say, well, what we actually care about is not predicting on held out chromosomes. What we care about is variant effect prediction. So then we should look at like chromatin accessibility QTLs, and hopefully they're less influenced by the bias. But yeah, it's a good question. Deep learning models are really good at picking up on artifacts in your data. There's all these like great examples from imaging, right? The one with melanoma detection, where it's like the melanomas had been circled with like a black pen, and the like non-malignant lesions hadn't. And so obviously the deep learning model is very good at like noticing, oh, there's like a circle around these. So yeah, you definitely have to be really careful with that stuff. Great. Okay. Yes, please. Yeah.
Can you rare variant association tests across the genome, incorporating the functional information? Use ABC model to map the CREs to the gene, right?
Right.
From my experience, it's very noisy in terms of mapping. Like,
I think it's probably pretty noisy. I'm not sure we have a great workaround for that. I mean, there is other data I'd like to try incorporating, say, looking at, there's all this single cell data now, right? And in principle, there's various models. I've actually had students in my class work on these models where you try and use the multi-omics data, where you have single cell attack and single cell RNA-seq, and then you can try and train a regression model that predicts expression from chromatin accessibility, and use that to try and say which chromatin peaks are related to gene expression, which is, at least it's very orthogonal to using Hi-C, which seems nice. I mean, in an ideal world, you'd have like an EQTL in every peak, and then but like EQTL data is really sparse. There is also, I mean, someone should probably be doing like a massive CRISPR-I study where they just like CRISPR-I every enhancer and see which genes get knocked down. So I don't know if NIH wants to fund like a $10 million grant or something. I don't know how much that would cost, but and I think microglia are like a somewhat difficult cell type to work with in the lab as well.
maybe you need monocytes.
Yeah, just do it in monocytes. Yeah, I mean, probably that would be pretty good already.
But there are also data sets of activated monocytes that
that's true.
Right?
Yeah.
It would be interesting to see actually if activated monocytes give you like better LD score regression.
Yeah. Tafiq's lab have done some stuff of trying to activate microglia as well, actually, and then do their RNA-seq. Yeah, so that's a good point of like, and actually, I think even actually in the grant proposal, we had some stuff about like we should train the deep learning models on like activated and activated states, because I know there's an argument with the immune cells that you're only really seeing their like true colors when they're activated. Yeah, it's a good
At
point.
least
Was
you know
more
what
things
cell
to.
type you're going after.
Yeah.
For many other
Right.
diseases.
Yeah, that's true. Yeah, to a big extent as well. Yeah, it's probably not all microglia, to be clear, but like, yeah, great.
Thanks.
Great.
That's great.
Thank you all for staying late.